Hey guys, in this video we'll study the restriction enzyme. Basically, restriction enzymes recognizes a particular target sequence in a duplex DNA molecule and breaks a polynucleotide chain within the sequence to give rise to discrete fragments of defined length and sequence. In simple words, restriction enzymes are bacterial enzymes that cut DNA molecules internally at a specific recognition site, and that is why these are also known as molecular seizures. Re- Restriction enzymes were first discovered and studied by Arbor, Smith, and Nathan. Haemophilus influenzae was the first restriction enzyme whose DNA sequencing was done and was discovered by Hamilton Smith. The restriction enzyme isolated from this bacteria was HIND2. All the restriction enzymes have specific recognition sequence within the DNA, and these are generally 4 to 6 base pair in length. These recognition sequences are often called palindromic sequences. For example, GAATCC, GGATTC. It appears identical when read from both forward and backward direction. Each individual enzyme recognizes only one specific sequence. For example, for EcoR1 it is GAATTC and for BAMH1 it is GGATCC. But sometimes the same sequence may be recognized by two or different restriction enzymes. Such type of enzymes are known as isosomers. For example, SAMA1 and ZAMA1 both have same restriction sequence, that is CCC-GGG. Now let's discuss about the nomenclature of the restriction enzyme. The uniform system of nomenclature was proposed by Smith and Nathan. Let's take an example for this. For example, EcoR1. This restriction enzyme is isolated from bacterial strain Escherichia coli, in which E denotes the first alphabet of the genus, CO denotes for the first two alphabets of the species, R is referring to the strain of bacteria, here it is RY13, and 1 is just a Roman number indicating that it was the first enzyme to be isolated from this bacterial strain. As restriction enzyme cut DNA molecules, so the restrictive host must of course protect its own DNA from the potentially lethal effects of the restriction enzyme, and so its DNA must be appropriately modified. The modification involves the methylation of certain bases. It is a normal restriction phenomenon, where a restriction enzyme cuts DNA molecule at a specific site and produces different fragments. As we see, it is an unmethylated DNA and modification enzyme, that is DAM methylates, that adds a methyl group to one or two bases within the sequence recognized by the enzyme. Once a base is modified, the sequence cannot be digested. Here, EcoR1 will not cleave the methylated DNA. In this way, host protect its own DNA from restriction enzyme. Let's stop today's video. In the next video, we will discuss the type of restriction enzyme and different types of ends produced by restriction enzyme. Thank you.